We're going to talk about different hormones and their roles in metabolism. So, the first part of this PowerPoint I'll let you read on your own. A lot of it's review from the discussion of G protein cascade that we had previous. And then there's some on the food pyramid, which you will get a lot more information in your nutrition and dietitian classes. So, let's go ahead and talk about some of the key enzymes. One was epinephrine, which acts on muscle tissue, and its goal is to raise levels of glucose. Glucagon is a peptide chain. It's released by alpha cells in the pancreas, and it acts on the liver to increase the availability of glucose. And then insulin is the opposite effect. It's there to ensure that glucose does not reach an excessive level within the blood. And so it will be triggering the intake of glucose by different cells. So epinephrine is a very similar looking structure to tyrosine. And so it's actually derived from tyrosine, so very similar. And it's released from the adrenal glands in response to stress. And what it's going to do is help activate the release of glucose for more muscle contraction. So this is a pathway that we already studied. It just has a different look, kind of nice looking actually. It's the idea that epinephrine will bind to a receptor. It stimulates the G protein, which activates adenylate cyclase to convert ATP to cyclic AMP, and that increases the activity of protein kinase. And then protein kinase does two things. It stops the synthesis of glycogen from glucose, and then it creates another series of phosphorylation events that ultimately break down glycogen into glucose. So the point is to release glucose. So this is the same signal transduction pathway we studied before. It's just in a different look of a slide. Okay, now in the liver, glucagon is going to do two things. One, it's going to increase gluconeogenesis. And it's going to halt glycolysis so that glucose is not used up by the liver so that it can free it into the bloodstream. Okay, so the same type of G protein cascade occurs, except this part here is different. We had not seen this type of regulation. It has more detail than I think you need to know. The one thing I want you to realize, though, is ultimately, do you see down here, these are two enzymes that we talked about, phosphofructokinase and fructose bisphosphate. So this is the PFK that we see in glycolysis. And then this is fructose bisphosphate is the enzyme in gluconeogenesis. Remember, it has several different enzymes that are used to reverse the process, and in those steps in glycolysis that were a very negative delta G, they can't go backwards, so the enzymes are different, and that's one spot, right? Because phosphofructokinase, step 3, very exergonic, so the reverse process is a different enzyme. In this case, there's a molecule that I didn't, it actually was in that PowerPoint, but I didn't focus on it, which was fructose 2,6-phosphate. And it's okay. I don't normally ask that, but all I want you to understand is you still have this G protein cascade. You have some new enzymes here that we hadn't discussed before, but the ultimate goal is to decrease phosphofructokinase's action and then that suppresses glycolysis. And why would you want glycolysis suppressed is that the liver won't use it for energy so that it can divert it to the bloodstream. Okay, and so this enzyme then, I'm sorry, this hormone is in response to low blood sugar. Okay, and then at the same time that glycolysis is suppressed, gluconeogenesis enzyme is activated. And again, gluconeogenesis is to make glucose. So this is an important discussion. It's just you don't need to know all the nitty gritty here in the middle of this diagram. So it's still a G protein cascade, but the ultimate goal is to regulate glycolysis, deactivate it, suppress it, and increase gluconeogenesis. Okay, so that's the written part to that. You can read that on your own leisure. I summarized it. All right, so for insulin, insulin is in response to higher blood sugar. So after a meal, it's a peptide hormone. It's secreted in the pancreas. Its active form is a 51 amino acid peptide. It has an alpha chain and a beta chain, and that are held together by disulfide linkages. Okay, and its precursor is called proinsulin. It's interesting here just to kind of give you a feel for the fact that these hormones all look a little bit different from each other. It's kind of interesting. And so let's talk about what insulin's going to do. It's going to bind to a receptor site on the cell membrane, and it's going to trigger a beta subunit to phosphorylate a tyrosine residue on the interior portion. Okay, and then the receptors, once it's phosphorylated, it will target proteins called insulin receptor substrates. And those guys are the secondary messengers. So this is a slightly, or not slightly different, it is a different signaling mechanism. So here's the insulin bound to the alpha subunits that create an autophosphorylation in the tyrosine kinase domains. Okay, now I don't normally ask about this receptor, but it's our theme of the course is how signals are sent. And one of the biggest themes that we have is phosphorylation, right? And the other part that's important here is that a discussion about insulin receptors plays a big role in type 2 diabetes. So we'll get to that here in a second. All right, so here are all the things that insulin does on metabolism. Highlights, so I won't read all of these, but some highlights is that it promotes glucose uptake in muscles, and the target is the GLUT4 transporter that we talked about. Okay, so that's important. It activates in the liver glucose breakdown, okay, through glycolysis. So that glucokinase is actually like hexokinase. So that's going to be increasing its function. So it's going to promote glycolysis. Okay. So I'll let you read some of these other ones. It in the muscle and liver, it will increase acetyl-CoA production, the pyruvate dehydrogenase. Okay. Oh, one other thing, it's going to produce glycogen synthesis. So this is interesting because these were the two processes that we looked at with epinephrine. So it's the opposite, which is that you're going to promote glucose to go to glycogen, and you're going to shut down the breakdown of glycogen because you're trying to pull insulin's pulling glucose into the cell, but then inside the cell, it has to do something with it. Okay. All right. So, as nutritionists and healthcare providers in general, it's really important to understand diabetes because it's such a national crisis. And type 1 diabetes is where a person does not have the ability to make insulin, and the beta cells of the pancreatic cells um, are compromised. And so, what will happen is that you just don't have enough insulin. This is usually diagnosed in early in life. And so, these are people that have to inject themselves with insulin since they were children. Um, type 2 diabetes is where there's a issue with the receptors that we just looked at. That signal between hormone and receptors is not working. Okay. And type 2 diabetes tends to start later in life, people that tend to be obese. So it's the idea is that the insulin's there, but there's a breakdown in signal transduction. Okay. I won't go into this particular slide. We don't have time to do that, but certainly there is significant research on type 2 diabetes and what is the exact mechanism that causes it. A lot is still, after all these years, unknown about that process. One interesting note is that sometimes type 2 can be regulated through diet and exercise, but sometimes it can't. And usually, individuals that can't get it managed are given insulin. So it's kind of an interesting thing. Okay, so insulin's primary job is to stimulate those glucose transporters, the GLUT4, to take up glucose from the blood. And then the other thing that it does is it says, hey, we don't want to break down glycogen, right? Because we want to keep glucose inside of the cell. So it turns on glycogen synthesis. It stimulates glycolysis in the liver and muscle, meaning that once it's inside the cell has to do something with that glucose. It turns off gluconeogenesis, and it stimulates fatty acid synthesis and storage. Okay. All right. So these next couple slides, there's only a few left in our class, but they're really important ones. It's kind of pulling together all of this. So we have a well-fed state. That means that we have a high amount of blood glucose. We've just eaten. We have eaten glucose, fats, proteins in a well-balanced meal. Okay, the high amounts of glucose trigger a response in pancreatic cells that has its own separate pathway that we normally don't discuss, but I want you to be aware that it's the glucose that stimulates through a whole series of pathways the release of insulin. Okay, now insulin, let's start. It's going to bind to the liver, and in the liver, it's going to promote glucose to glycogen. So in other words, I think I just said it earlier, that once inside the cell, it has to go somewhere. So one, glycogen it gets stored. Another was that glycolysis is activated, form acetyl-CoA. If you are active, right, or there's an energy demand, now this is not the muscle contraction because we're in the liver, but so the liver has to make ATP for its own metabolic processes. So this would be Krebs, right? Electron transport. Okay. And then it will make fats. So just going to make a little side note here. Do you recall that there was the idea that we take in fats, we eat them, we process them, we have stored fats, and then here's the other idea is that we make fats in the liver as well. Those guys have to get transferred to adipose cells, and here's the very low density lipoproteins that will take it and get stored in adipose tissue. This is showing also a transport to the muscles for beta oxidation if there is a demand for that. Okay. All right. The fats that we eat in a well-fed state, we talked a lot about how they go through all that process and ultimately chylomicrons, right, will transport it to adipose tissue. Okay. The other thing too is that glucose, what happens to glucose? Well, glucose, all right, will go to the brain as the primary fuel for the brain to produce ATP through glycolysis, citric acid cycle, and electron transport. So most of the sugars that we eat go to power the brain. Okay. This also shows some of the glucose being delivered to the liver to be stored as glycogen, some of the glucose being taken to the muscle, right? Because the muscles will also store some glycogen, and then you hopefully will be active in some way, so you have a demand for energy in the muscle to take glucose through our pathways of cellular respiration. Lastly, amino acids that we eat will go also to the liver for processing, and it will go into tissue protein, right? The carbon skeleton will be used and reshuffled around the body for different enzymes and key molecules like the ones in Krebs cycle. And then the ammonia part will ultimately make its way through urea cycle. So our well-fed state is important because after we eat a meal, all our key players, glucose, amino acids, and fats, will play a role. But I want you to notice, really important, that amino acids are proteins really in the well-fed state will be for tissue protein. If you notice in this diagram, it's not concerned with them as an energy source because we would be relying on glucose and fats. Now, glucose is going to be our number one go-to because it's water soluble. It can get to the brain easy. It can go to the muscles. But if you also notice, excess glucose will make its way to form fats right through this pathway here. One other thing, there's one noticeable thing, is that fatty acids, they don't go directly to the brain as a fuel because they don't pass through the blood-brain barrier. So there's no arrow into there for fatty acids. Okay, so the fasting state. So after about 4 to 6 hours, but let's just say 6 hours without eating, we start going into the fasting state. And then there's a progression. You could go to the 12-hour mark, 24-hour mark, 2-day mark. So what I'm describing here is the idea that after 6 hours, things start to shift. But as you keep progressing in the fasting state, the processes I describe on this slide will get more prevalent the longer you go. Okay, so in this case, the pancreas will excrete glucagon. Glucagon then will bind to liver receptors. And what's going to happen is the following, that the cell now is going to be concerned with releasing glucose. So that's going to be freeing up glucose. So glycogen is going to form glucose 6-phosphate, which will then go to glucose. And then that's going to be through the bloodstream, go to the brain to help power the brain. All right, now the other thing is that glycolysis, okay, glycolysis is going to be shut down and limited. So you see a decrease in glycolysis, and you start shifting towards gluconeogenesis. Okay, so again, this is starting at this approximately the 6-hour mark. It won't be fully gluconeogenesis at that point. So it begins to shift over to that. And then as you keep going on your fasting state, you get deeper and deeper into this. Okay, so gluconeogenesis becomes more important. And that would mean the conversion of pyruvate to gluconeogenesis. Now, the main things that can be brought in for gluconeogenesis are pyruvate, glycerol, which we'll talk about in a moment. Start seeing amino acids that are glucogenic, which were a lot of them. Okay, so pyruvate, instead of going through glycolysis, can form glucose, gluconeogenesis. See this glycerol? Okay, now what's that coming from? That's because, remember, our triacyl glycerols are made up of 3 fatty acids and glycerol. So the fatty acids, those get split apart by lipases. The glycerol will ultimately end up in the liver, and it is a good fuel for, it also can be converted to glucose through gluconeogenesis. Fatty acids will go to the liver, and remember, they can undergo beta oxidation, but that means that you'll see an increase in acetyl-CoA. And then that increase in acetyl-CoA will make ketone bodies, which then are an alternative fuel for the brain. Amino acids then become important because you have some of them that are glucogenic. Not shown in this picture would be the ones that are also ketogenic, which means they form acetyl-CoA to make ketone bodies. Ketone bodies can also go to the muscles as an alternative fuel there as well. Interestingly enough, the muscles then can use them for energy. That is not an ideal source of fuel, though, for muscle contraction and in general. So, in other words, the way that I think of that is, you still need to walk around, you still have muscle contraction, but this is not meant to be after 6 hours in the fasting state. This is not meant for you to go run 5 miles, right? That's not the main role of it there. It's an alternative fuel for muscle contraction. Notice that muscles, proteins can be diverted from amino acids for gluconeogenesis. So really the role that we're seeing here is an important role of the liver in regulating blood glucose levels for the brain. And everything starts to shift towards protecting the brain. Now, when you have prolonged fasting or uncontrolled diabetes, everything that we started talking about is still in play, only it becomes a more extreme event. So proteins are degraded to make glucose. So you have glucogenic amino acids. The more those are broken down, the more that the more urea, and then that's exported to the kidney and excreted in urine. Okay, so that event's in the kidney, but this is a hepatocyte, so this is a liver cell still. I just switched it from the liver looking picture. Okay, then here comes the important thing that we've talked about, which is that everything's going now to glucose. Oxaloacetate is diverted for gluconeogenesis. And so now what you see is a massive shutdown of glycolysis. Okay, now in the previous slide, let's go back for a second. It didn't show that, but that's also at play here. Even when you're in just regular fasting state, you're going to slow down Krebs cycle, which is why you're forming ketone bodies instead. So you're still slowing Krebs cycle down. It's just in prolonged fasting, when you're looking at 12 hours, 24 hours, a couple days, you're really starting to pull everything for gluconeogenesis and glucose. So the glucose goes to the brain. Fatty acids, again, start to break down for acetyl-CoA. And because they can't enter into Krebs because oxaloacetate is being diverted, then they will form ketone bodies, which also form brain fuel. Okay, so the prolonged fasting is similar to the 6-hour mark. It's just more intense. Okay, and uncontrolled diabetes, when you don't control your diabetes, what happens there is that your blood glucose is high. It can't get into your cells though, so the liver cell starts to function like it's starving, right? Like it's in a fasting state, even though the bloodstream has it. So, in other words, in uncontrolled diabetes, you still have all these processes going on. You have protein degradation, fats get pulled out to make acetyl-CoA, but you can't run through Krebs. And since Krebs shuts down, you have an increase in ketone bodies. And then these guys are now in the blood. But in uncontrolled diabetes, you see such a large increase in ketone bodies that, remember, some of them are acids, carboxylic acids. So you see a decrease in blood pH, which is the ketosis. And that sometimes in uncontrolled diabetes goes to such an extreme event, blood acidosis, that creates a coma where people can die from it. So it's a diabetic coma. The blood buffers that we talked about, remember we started our journey together by talking a lot about the bloodstream biochemistry. There's so much acid in the blood that it can't compensate. So the blood buffer system that we looked at the beginning of the semester has trouble compensating for that. Okay, so that gets messed up. Okay, so let's see if there's anything else that I'm missing as I look at this. Okay, so one last thing before we move on or finish this up is when you are looking at diets, then diets that as a nutritionist or healthcare provider, then you really need to have your facts about these diagrams because they're really the heart of how we process things. So if I go back, the best diets will have a well-fed state where you have a well-balanced meal where you don't have ridiculously high amounts of any of them and you have a balance of them. And so really the body is meant to operate in this well-fed state where proteins are used for tissue protein, where you have glucose going to glycogen, you have glycolysis, you have some stored fats. It's just that the diets that people have now are so heavy on all of them that the arrows really pushing this way towards fat storage. But I hope that you see that carbohydrates, glucose is not a villain in our body. It is meant to really help power our brain and muscles contraction. It's just that if the percentage of sugar that people are ingesting is too high, then it just throws all of this into storage. One side note is that, and I didn't mention it at the time, that fructose can also enter into glycolysis. And that's a very hidden, not so hidden actually, sugar that then increases the amount of sugar that we ingest. So the high fructose corn syrup and added sugars, but if to a lot of food. But if you have a diet that is strong in fruits and vegetables, then you're really going to be ingesting glucose that's needed. It's just we don't need all the other added sugars in our life, okay, in our diet. When you haven't eaten for a long time, you switch to the fasting state. People like this idea of the fasting state because you pull from adipose. So I'm not going to lie to you that you will get thin if you don't, if you don't eat a lot after 6 hours, you start to pull from fats. But the problem here is that this is not conducive to being an active person because your muscles are really meant to run off of glucose. And secondly, everything here is the shift, if you notice, is not really muscle contraction being optimized. The shift is to the brain. Okay. And so you can really see in all these arrows that there's a shift towards protecting the brain. As a result of that, you will lose weight, right? but would it be in a healthy way? So diets that are completely devoid of carbohydrates will get you into this state. So you will be pulling from fats, but they're very difficult to maintain because your brain is always then running off of ketone bodies as secondary fuel. And they're just difficult to maintain for people in general because really the body wants to be in this state. That's the natural way for it to operate. And then in the extreme event here, where you really start to put a strain where you completely are shutting down Krebs cycle, prolonged fasting obviously then would not be the best route if you're trying to be active, because you are not going to be promoting glycolysis. Everything's going to be shifting. If you notice up here, brain, 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 right? The goal is to protect the brain. So really, from a biochemistry point of view, eating a well-balanced meal where you're exercising and you're not ingesting too much of any of those glucose, fats or proteins, excess is not a great thing, right? And so this is really the best way to maintain the body from a biochemistry perspective, where all the key players are doing what they're designed to do. That glucose is prevalent or important for the brain, in muscle, that you have some stored fats. Proteins are involved for tissue proteins. Okay. And okay, so that's pretty much it. We'll talk more about this in the Zoom discussion on Monday. And I'd like you to think about your diet. How are you approaching this? And it'd be nice if you could share some of that on Monday. And no judgment will be passed on it. It's just, it's good for us to talk about that. One last comment that diets like there's a popular like Mediterranean diets where you do have glucose, you do have fats, but they're unsaturated. You do have lean proteins. Like those do tend to fall really well into this discussion here of this slide. Okay. What doesn't fall well in here would be diets with all sorts of junk food, right? Where the glucose levels are too high, where the fat's too high, right? Because then what you'd see is this arrow for sure, because you're going to have too much glucose. So you're going to go this way and that way. And then the stored fats as well. The fats we ingest would come this way too. Okay.